Hi, my name is Nikhil Rao, and I am with 10x Genomics, and I am the commercial lead for the Visium Spatial Gene Expression platform here at 10x Genomics. Today, I'm hoping to convey the importance of why spatial information with gene expression is important, and also help you understand what is the product that we are in the middle of launching, and hopefully give you an idea of what that could do for your science. So we always like to start with this first slide, which is a quote from David Craig out of USC, which says, "Everyone is waiting for spatial. Without context, you can only learn so much." And we think that this is rather important because at 10x, we have been really the leader in single-cell gene expression and single-cell assays, which tells you the what to what's going on in a single cell. But we believe that this new technology can tell you the where to where genes are expressed on tissue samples that are really important to understanding the biology that you're interested in. And we think that there are a lot of groups and researchers really waiting for this technology, so we're really excited to start talking about it today. So why would you want to do what we call spatial analysis, and what is spatial analysis? Now, spatial is essentially overlaying gene expression onto an H&E image. There is the ability to basically section tissues and then look at gene expression globally within that tissue and localize it to areas that you might be interested in. What this does is it allows you to do gene expression without any kind of tissue dissociation. So in previous methods require you to RNA isolate out of the tissue, which means that you lose the integrity of the tissue and you don't know which RNA molecule came from what area of the tissue. So you're still able to retain the organization and tissue of the cellular microenvironment, but it also provides a way to look at cell types in the context of heterogeneous tissue based on gene expression. So you can identify cells based on whatever genes are expressed in those cells and understand where they're co-located within the tissue. And in this example here, you can see a pathologist has actually annotated a region of this tissue as cancerous. And in the subsequent panel that's in the fluorescent channel on the right, you can see that we use spatial here in this publication to detect the lymphocytes or the white blood cells that are really infiltrating the tumor, and the pathologist looked at that spatial information and said, yes, this is yielding a good prognosis because the tumor has a positive immune response. And B, there's similar annotation has been made by a pathologist where they say, oh, in this region there's cancer, but if you look at the spatial gene expression signature for lymphocytes, you can see that the immune cells are blocked from entering that cancer. So it gives you an extra layer of information that you wouldn't have been able to achieve with other technology methods. And this was deemed a poor prognosis because of the fact that you are not seeing a positive immune response. Now, you might say you might be able to do this with immunohistochemistry or other types of technologies, but the reality is that there are not an extensive number of markers that identify every cell type of interest. You may not know what cell types you need to be looking for, and our spatial platform allows you to look without sort of a biased view at everything that exists in the tissue section based off of all the mRNAs in the cells that exist in the tissue. So we believe that spatial has a place in most areas of biology, and that ranges from oncology, which is sort of the example that I just gave you, really important for tumor heterogeneity, tumor microenvironment, and white blood cell migration, as well as immunology, understanding immune populations, what are the types of immune cells, is it an M1, M2 response, all the way into developmental biology from mapping specific organs, as well as understanding development as a function of time and space, which is really kind of an emerging field. It also has a lot of applications in tissue engineering and wound healing, followed by neuroscience, in which groups are very interested in understanding the anatomy of the brain and studying neurodegenerative diseases. And we also call out pathology here, even though it's not really a research area. It is certainly a technique and a field in which a certain standard has been generated over the last years to study these types of research areas in tissue sections, but we think that this could enable pathology to do more than what it's done before, to provide an orthogonal method by which they are able to diagnose patients as well as treat patients based off of gene expression, not just morphology. So now I'll kind of transition into what is the product that we are talking about today and how is that going to help your research. So we have something called Visium, which is a platform that is sort of parallel to our other platform called the Chromium. However, Visium is not an instrument. It is a technology that we're using called the Spatial Gene Expression Solution that essentially allows you to look at unbiased, as we call unbiased, meaning looking at all the mRNAs that exist in the transcriptome, and high-throughput gene expression analysis for intact tissue sections from a wide variety of sample types, and we'll talk more about what those sample types are. But you can look at the biological architecture in normal versus disease. You can look for new biomarkers and specific cell types and regions within the tissue. But the big two bullet points that I like to hit here is that it really does integrate easily with current laboratory methods and tools. So again, we're not selling or providing an instrument. We are providing an assay that is really built such that it works with the infrastructure that you might already be familiar with or exists in the institution that you likely work in. And we'll get to what that infrastructure looks like. And the last point is that this is really a one-day end-to-end workflow. From the minute you section your tissue to getting a final library ready for sequencing can be done easily within one day. And in general, we use slides as well as reagents here to provide you all that has the ability to look at very much sort of a high sensitivity assay that interrogates thousands of genes detected per spot, and we'll talk about what the spot looks like. The resolution is extremely high with 5,000 spots per capture area. This is where you place the tissue section on the slide, and the spots represent the captures of the mRNA molecules onto the slide. Each spot is 55 microns in diameter, and we have a very close packing of the spots. Again, everything is unbiased, so it's a poly-A-based capture method. It's efficient. It's a one-day workflow time. All the slides and really reagents really important for our kit are included. And in general, we see that our cell resolution is anywhere from 1 to 10 cells per spot, meaning that for 1 to 10 cells, we're able to look at that regional gene expression that's sort of an average of those cells. So depending on the tissue type, you might have one cell that gets interrogated and you're basically getting single cell resolution of gene expression. But if a tissue has a lot of dense cells closely packed to one another, you might get upwards of 10 cells per spot that kind of average out the gene expression in that area. In any case, you get to really get a holistic picture of what's going on within the tissue, and I'll show you that via data and images in the next few slides. So you might have seen that we've had a technology that has existed on the marketplace to date, and that's the spatial transcriptomics technology that is really a company that 10x had acquired last year. And this technology was incredibly impactful to the field. And this consisted, again, of these spots that are on a glass slide where you put your tissue section. And here you can see an image of a mouse olfactory bulb in which the olfactory bulb was placed onto the capture area. And you can actually see anatomical features based on the heat map that is showing the unique molecular identifiers or the number of molecules picked up in that generalized region. Since that technology has been out on the market, we at 10x have done a great deal of work to simplify the workflow and also improve the performance. And you can see now with our new Visium spatial gene expression platform that the resolution, the density of the spots, as well as the ability to identify anatomical features in this mouse olfactory bulb has greatly improved. And you can see there that we now see about 80,000 UMIs versus 14, so we've really improved the sensitivity of the assay dramatically. So what does this workflow look like? At a high level, we have some sample prep steps that involve sectioning tissue onto the slide. We then will take this slide, once the tissues are on, through to H&E, so you actually stain the sections and you can image in bright field. We ask you to essentially take a stitched image of the entire capture area, which are those 4 boxes there, to save for later as a TIFF or a JPEG. Then you would take the slide and do an RNA isolation followed by cDNA synthesis and library construction and run on a sequencer. Once the sequencing is finished, we import the sequencing data along with the image that was taken from the slide into our data visualization tool to allow you to pair gene expression with the image analysis. So there's a lot of various steps involved in the workflow, and we'll get into detail on what those look like. Looking at the slide itself, I mentioned that there are 4 capture areas on the slide. Each of those areas has the 5,000 barcoded spots that are at 100 micron center-to-center distance. And then each of those spots contains millions of oligos or barcodes that you can see on the right that have a partial read 1 for sequencing, a spatial barcode, which is essentially a sequence that allows you in sequencing to understand where the molecule came from on the slide. So instead of in our single cell assays, which will have a cell barcode that will relate all the sequencing data to a specific cell, in this case, we have a spot barcode or spatial barcode that relates all the sequencing data to a place on the slide. A UMI, which is a unique molecular identifier again, which basically counts the number of molecules, and a poly-DT, which captures the mRNA. And you can see here a blow-up of one of those capture areas, and it is a square shape that in the black regions contains all of these barcoded spots, and that is a 6.5 by 6.5 millimeter square area. And on the outside of that, you can see these red dots, and what these red dots are is what we call a fiducial frame. What that is a larger dots that don't contain any kind of spatial information, nor do they pick up RNA, but they're used in image registration, so that when you take the image and you pair the sequencing data on top, the sequencing data basically finds its way to the right spot on the slide based off of where the image is taken and how those fiducials show up in image analysis. So it's basically guiding the sequencing data to the right guideposts so that it maps perfectly onto the slide. And so all it is a way for us to very accurately map the gene expression data onto the image data. And so now you can see we have sort of a more granular workflow here where, again, you would use a cryostat to section the tissue block onto the slide. Then you would stain an image, so you would do an H&E staining followed by imaging, and then you would permeabilize the tissue such that the mRNAs fall out of the tissue onto the oligo-DT, convert the RNA to cDNA on the slide, and then at that point you can take everything off the slide and put them into tubes and do sort of PCR-based library prep for sequencing. So the kits that we provide are the tissue optimization slide and reagent kit, as well as the gene expression slide and reagent kit. And I mentioned that there is a step where we permeabilize the tissue. Now, for every tissue, you might have a various degree of permeabilization. You can imagine that some dense tissues might take more time to permeabilize. Some that are more acellular or maybe less extracellular matrix heavy are actually easier to permeabilize. So we give you a kit that allows you to, for any individual tissue, optimize those conditions. This is that tissue optimization kit in which you have these 8 capture areas here as the image shows, but they don't include any spatial information. It is purely to optimize the tissue optimization. Once you understand and can isolate the maximum amount of mRNA based on a specific condition for permeabilization, you take that condition into the next kit for the spatial gene expression slide and reagent kit. So in this kit, this is where all the spatial analysis happens, and you're able to pair the gene expression with the image data. So really the tissue optimization kit we see being used as sort of a one-time use for any given tissue type. And once you really find that optimal condition, you just use the gene expression slide reagent kit from there on out for a given study. We've tested a large number of tissues internally on Visium, ranging from human, mouse, rat, and in previous iterations, plants and insects have also shown proof of concept to work. These tissues haven't seen any issue with testing them. There's no limitation. And we believe that these tissues internally, these lists will continue to grow. This is not an all-inclusive list, as we just haven't had the bandwidth to test every tissue out there, but we have plans on testing a large number of tissues in the near future. And our software is such that it's license-free, and it's very similar to what we provided with our single-cell portfolio, which is a software called, you might be familiar with, Cell Ranger. For spatial analysis, we deem it Space Ranger, as well as Loop Browser for our image analysis. So again, providing automated image processing and interactive data visualization tools to kind of be able to use as if you had no kind of software expertise. It's actually very straightforward to use, even if you're not a bioinformatician. And with these two software tools, you can really do a lot of different analyses that I'll kind of go into detail on. But in an example that we like to show just to compare with single cell is that you can look at an unbiased TS&E plot based off of gene expression and look at the clustering of these cells based off that gene expression, and then annotate directly back onto the image so that you can see where those cell clusters fit onto the slide. So you're taking things that are based on genes in space and putting them into a location on sort of an H&E map. So show you some applications of how this is used and really kind of specifically talk about the mouse brain. The mouse brain is something we've done a lot of internal work on, primarily because it's really easy to see the anatomy via H&E, but also has a really diverse set of cell types that cluster really nicely together based on the anatomy alone. So you can see in A, we have an H&E of a mouse brain, and in B, we've done spatial and asked the software to tell us how many unique molecular identifiers exist in the region of that H&E. And you can see that in certain areas, there are more UMIs picked up than in others. And then we ask the question of how many genes, unique genes, are identified in any given location. You can see in the section C here that there's a lot more genes sort of on the periphery of this tissue than in the center. And then you can go further to say, okay, knowing that there are different levels of genes in each of these cells, can we create populations of cells that are very similar to one another based on gene expression? And this is sort of in the previous slide what I showed with the TS&E plot, but mapped onto the actual H&E slide. So you can see here we have 8 clusters, and then you can even go further and say, hey, I am interested in cluster 4, the green cluster, and these are the 10 or so genes that are highly expressed in cluster 4. So you're able to look at any individual cell cluster and I understand what is the gene expression within that cluster. Now, to show you what it looks like in our software really does a good job of making this super user-friendly from sort of a GUI interface. So you can see here on the right column, you have your 8 clusters. You can select or deselect them. And on the bottom, you can see the genes per cluster that give you sort of the p-value of each gene in a given cluster. And you can also mess at the top with the spot opacity. So you could take away the spots or you could include the spots depending on what sort of visualization you like to see on the tissue section. But kind of moving even further in the software, you can zoom and get really kind of high resolution understanding of a local region within the brain. And here we've zoomed into the top left portion. You can really resolve what those spots look like and what spots cluster with what, which population in this example here. Now, you might be interested in one specific gene, and in this example, we show with the mouse brain that there are two genes in particular, TMSB4X and selenau, that are expressed in the hippocampus. And so we knew via literature that these two genes would be highly expressed in the hippocampus. And you can see that with spatial, when we select these two, that they are indeed confirmed in that anatomical region of interest. And what's nice about this is it sort of provides a positive control, so if you're ever in doubt that, you're identifying a gene in a region that's very interesting and you don't know whether it's true or not, you could always look at a gene that is anatomically relevant and making sure that it actually does locate in the vicinity of where it should be to give you a kind of a positive control reinforcement that what you're seeing is true. And so you can actually identify and zoom into those regions and confirm that these genes are highly expressed. And in the software, this is what it looks like. You can change at the bottom right your color scheme as well to make it easier to visualize. But this gives you an example of SPINK8 and DDIT4L, which are genes that are expressed in the brain tissue but in different regions, and just gives you another idea of how you can look at individual genes on this tissue. The last feature, which is incredibly impactful, is the ability to look at anatomical investigation. So you might have been familiar with other technologies that allow you to look at regions of interest within the tissue section. This software tool allows you to do that on the backside in the software analysis. So you've gotten gene expression globally of the tissue, but you may only be interested in, let's say, the top right region. Maybe that was deemed by a pathologist as cancerous, and you want to say, hey, what are the genes in that region? I don't want to look at it based off of unbiased gene expression and clustering. I want to look at it based off of a bias that I have in that we believe that anatomy is important for our biology. So there's a feature that allows you to sort of select without sort of, predefined geometries what area you're interested in, and then again, pull out at the bottom the genes in that specific area that are of interest. And we think that this tool will certainly be of interest to a lot of the users that we've talked to that are eager to try this technology. And just to give you a sense of other places where this is used, since, neuroscience is one field, but also in cancer, and this is probably one of the more impactful studies that has been done with spatial gene expression, where this group, Berglund et al. In Nature in 2018, wanted to look at prostate cancer sections and ask pathologists the questions of, okay, can you annotate where on this prostate cancer tissue there is cancer versus sort of stroma and the pin glands? And then we're going to take this through gene expression workflow and assign 6 genes that we will call factor signatures that are highly expressed in cancer. And we'll perform sort of a heat map analysis of where those genes are upregulated on the spatial tissue. And when they did this, they saw 2 scenarios that came up frequently. In the top scenario, you see that the pathologist annotated a region of interest with cancer and pin glands. And in the factor activity map, actually got pretty good confirmation that there is cancer in that region based off of those 6 genes, as you can see in the heat map in the top, as well as sort of a positive control that stroma existed in the regions that are outside of that area on the far right. But in the bottom case, which is incredibly profound, is that the pathologist did not annotate this H&E section as being cancerous, but when spatial gene expression was performed and looked at those 6 genes, you can see that cancer was highly prevalent based off of gene expression in that area. Now, this obviously needs to be followed up, as there are 2 readouts that say differing things and probably need another orthogonal means to validate these. But what it does is it shows you that 2 dimensions might inform you better than the 1. And it also informs you that maybe you are missing things if you don't perform a spatial gene expression analysis on a tissue section. So we think that this is going to be really impactful, again, in oncology as well as in the pathology space. There's another example that was pretty impactful from Maniatis et al. In 2019, which studied ALS. And what they did is they looked at mouse spinal cords over the course of a disease model of ALS, but then they also correlated to human spinal cords post-mortem from actual ALS tissues, patients. And what they wanted to know is there something interesting about the interaction of these cells in pre and post-ALS onset of symptoms? So in the mouse models, you can imagine that before even witnessing symptoms, you are able to resect these spinal cords and look at them, and also post, as well as with the post-human brain, post-mortem ALS spinal cord tissue. And what they found in general, using only really spatial analysis, is that they could look at really defined local regions in which the microglia underwent some sort of dysfunction that really was before what you would consider an astroglial dysfunction, which is usually indicative of symptoms in ALS. And they found that these are very proximal to the motor neurons, indicating that that actually precedes the kind of the symptom, the negative symptoms that you experience in sort of the motor function. And so without the spatial, they weren't able to know sort of how these cells interact with one another and how they co-locate with one another. But what they might have actually found is sort of more of a predictive biomarker that occurs in microglial cells way prior to the onset of some of the traditional symptomatic conditions that most people are looking for. And sort of lastly, a great example, and we believe that a lot of our sort of current single cell researchers are really looking forward to using spatial for, is this idea of looking at single cell information and spatial information at the same time. So in the top here, you can see that this group out of SciLife has looked at a developmental heart tissue and done a dissociation on it to look at the single cell gene expression and clustered cell based off those gene expressions to kind of understand what are those major anatomical regions based on single cell information. But then on the bottom, they also did in parallel tissue sectioning on these developmental heart sections and clustered the spatial transcriptomic data in a similar manner. And then they merged the data types. So they said they want the resolution of the single cell, but merged to correlate with the gene expression of the spatial data. And what they were able to do then is trace single cells back to their spatial position and then actually three-dimensionally render the heart back into sort of a larger gross model. And so we believe that many people are working towards sort of these 3D molecular atlases. In this case, it was the developing human heart, but we've seen cases where people are interested in doing embryos, brain and spinal cord, as well as, you know, even intestines. And we think that really spatial is the tool that will exist to really for these sort of human cell atlas or human tumor atlas projects that are really going to kind of accelerate as a result of these findings. So lastly, you know, we are hopefully convinced you that, you know, this is going to be able to allow you to envision new dimensions by pairing gene expression, morphology, and protein with fresh frozen tissue samples. And we believe that, you know, this is sort of a more rapid, robust, streamlined workflow that can be easily implemented in sort of a typical lab. And if the infrastructure doesn't exist, there are places that easily kind of provide these as services that will enable most researchers to use this. And the pre-order for this solution is available today. You can go on our website at 10xgenomics.com/spatial and look up for more information. We are hoping to ship this product later this fall, closer to the end of October, early November timeframe. But get your orders in today if you're interested. And just so everybody is aware, you know, we have a pretty large and growing single cell portfolio. And now we have a spatial portfolio, and gene expression is sort of our first application. But we do envision that we will include more applications in the future, and we're eager to hear what are the applications in which you are interested in to kind of take advantage of this technology moving forward. So stay tuned for that. We do have a nice roadmap on the spatial side that is in progress today. So with that, I'm happy to take more questions after this presentation via email, and I'll try to get back to you as soon as I can. Thank you very much.